Craig.
Cool.
Eric.
Hi.
Hey,
Eric
wait a minute.
Bowman.
Wow.
I'm
With
trying
also
to catch up here. Wait a minute.
short hair. Wow.
Oh, yeah. For,
Oh, Vicky. Hey.
for my 50th birthday.
And Eric.
Hey,
Yeah, it's going to be a bunch of people.
excellent.
Hello.
Oh,
Hey.
Hi.
Hi, Matt. We
Hi,
got
Mark.
the two,
How are you doing?
Great. We got the two Wisconsin people here, Sarah and Matt.
Oh, Becky.
Great.
Yeah, it's roasting here today. I don't know if you're in a heat wave also where you are.
It's not too bad right now. It's 81.
That's not as bad. It's been like solid 90s for 2 weeks. We're in a drought.
Wow. Wow.
Oh, wow.
Yeah.
Hi, Kathleen.
Hi, Mark, and hi, Matt.
Hi, Kathleen.
Hey.
Hi, Becky.
Hi, Mark. How are you?
Good. I don't hear the British accent. How's your British
Oh,
accent?
it will appear awkwardly in a minute, I'm sure.
Okay, that's
So,
good.
Hi.
Great to see you.
Yeah, thank you for having me.
Yeah, that's G. Yeah,
Oh, gee, all the way from Thailand.
yeah.
What time is it in Thailand?
Oh, Meredith.
Oh, Meredith, hi.
Hi,
Hi.
congratulations.
Thank you. We'll all go around later and introduce ourselves. Give a couple more minutes for a few other people to show up. I think we had at least 20
Well, we'll see.
people said yes. 4, 12, yeah. Good.
hi, Mark, Cindy, and everyone.
Hi, G.
I'm
What
from
time
Thailand.
G.
Yeah, what time is it for you, G?
It's 3 o'clock in the morning.
Oh my goodness. Oh,
Yeah, that's a committed scientist. That's
So
for
it's
sure.
You are
just
still
me.
working hard in the night. You are still working hard in the night,
Yeah.
like usual. At 3 o'clock.
Oh, Vijay.
Yeah.
Hi.
Hi.
I think VG knows the most people here, probably.
Oh,
Hey, Austin.
Hey, Mark.
How are
Hey.
you? Good to see you. Glad you could come.
Yeah, good to see you
Hi,
too.
Austin. Hi, Austin.
who's
This is Karthik.
Hey,
Hi.
Karthik. How you doing?
How are you? Good. Nice to see you.
Where are you?
I'm in Bay Area.
Okay. I'm in Virginia Beach.
Well,
Oh,
oh, nice.
so Mark, do you want to start?
yeah, I guess we could start.
Welcome everybody. Oh,
Well-
there's Tom. Oh, wait.
Oh,
Tom and Jim.
Tom Trans.
Tom.
Yes. Hi, Tom.
Tom.
Wow.
Oh, he's still connecting.
Yeah.
Oh, there he is.
Hi, Tom. Good to see you.
Hey. Hi,
How are
everybody.
you?
Good.
Wow.
Good to see everybody.
It's a great gathering. Well, I want to welcome everyone. There'll be more people coming. I decided to retire for no one specific reason. I thought I'd retire on a high point. And Cindy and I have plans to do a lot of different things. Personally, learning web development, try to get some new skills. Still have a couple of papers I'm working on, and we're planning on doing some traveling pretty soon. We're going to New Jersey, we're going to Houston, and then in October, we're planning on going to Israel for a couple of months.
Wonderful.
I'll be working at the Hebrew U in Jerusalem, like I did for my sabbatical. But I'm really happy everyone could come because all of you folks were so important to my happiness at U of M. It was great having you. So I thought we would go around and people could introduce themselves, maybe say when you were in the lab, if you remember. I don't. I don't remember the exact time. And then tell us a little bit about what you're doing now so we can celebrate things for you since you've been in the lab. So I thought we would start with G because he's the one who's probably going to fall asleep on us the first. So, G, go ahead.
Okay. Hi, everyone. I see Tom, Bart, and Eric, Craig, Richie, those who were around. Okay. And Daniel and Noam, you grew up so big. I couldn't remember you.
We should
Okay,
say
my
our
name is
son-
Chirandon. Everyone call me G, and I was in Saper Labs during 91 to 97. Okay, long time ago. I think I was there after Tom,
Yeah.
right? Tom was the first, and then Heidi, and then me, and then Craig, if I remember, and Bart. Yeah. Okay. Right now, I'm at the Department of Biochemistry at the Faculty of Science, Mahidol University in Thailand. Okay, just stepped down from being the head of the department
Wow.
about a year and a half. Okay, and getting back into my research. I'm still a crystallographer, so doing structure and some other things. Most of the things I'm doing is to try to mutate protein, trying to engineer something, and that's it.
Great.
And I'm sending you souvenir, Mark. Hopefully
Okay.
it reached you this week, if not delay further. Okay.
Hey, thank you. That's really nice. Yeah. Give our regards to Na.
Yeah, thank
Yeah.
you. She say hi to
Okay.
you and
Great.
Cindy. Yeah.
Good.
Yeah.
Couldn't convince her to stay up till 3.
Yeah.
Well, the question is, did you stay up or did you just get up?
I stay up to about 1 o'clock and then got slept a little bit and then woke up again.
Yeah. Yeah. So G was a graduate student in the lab. So I don't know what order to go, but I think I may just go the order on the screen here so I don't miss anyone. Karthik, do you want to introduce yourself?
Oh, hi everyone. It's very nice to see all the members of Mark's lab. Mark, happy retirement. So my name is Karthik. I was a grad student in Mark's lab during 2005 to 2010 December, and yeah, I overlapped with Austin and Vijay. And maybe I saw I came across all of your names in the lab, but then it's nice to put a face to the names. And yeah, so after graduation, I did my postdoc also in crystallography. I moved to the Bay Area, and right now I'm working for Merck in the early discovery programs. Yeah, having started out as a crystallographer, I still have my passion for it. But right now, I'm doing more and more of engineering, antibody design, and also doing automation, industry automation, and expression characterization, and more of the stuff that goes before seeing the crystals, I think.
Good.
Yeah, I had a really wonderful time with Mark's lab. It was, I still remember I got my admissions letter from Mark, and I still have it. And and yeah, it was, I was just trying to go through and remember all those fond memories. I remember the canoe trip in the Gallup Park, or near there. I forget all the names now, but yeah, I had a nice colleagues with Vijay and Austin. Austin was like my mentor in the lab, and he showed me the ropes. And I remember all those times I was setting up crystals in the cold room, shivering, and and yeah, Mark was very kind, and was very understanding and helpful. yeah, he allowed me to travel to India every year. And yeah, it's just, it feels like, I know G mentioned it's a long time. It feels like a long time for me too. Like it's more than 10 years now. And yeah, I owe a big thank you to Mark for giving me this career and giving me the start. And thank you a lot, Mark. I wish you great happiness in your retirement. And yeah,
Appreciated all the work that you did and all the companionship over all the years you were in the lab.
Yeah, there's so many memories. I think I should also mention our time in conferences when we traveled together.
Right.
How we lost our poster tubes.
Lost our poster, yes. Yes.
Yeah, there was so many memories. I'm sure we'll go around. It's just an introduction. So yeah, it's very nice to have this gathering.
Thank you, Karthik.
Yeah, it's been a very rewarding experience. It's a pleasure knowing you. I'm very sorry. I feel guilty that I haven't been in more in touch over recent years. But I'm very happy for you, Mark, and your family, and wish you the best. Yeah.
Thanks.
Yeah.
I think we'll move on to VG. VG?
Hi, Mark. Like Karthik was telling, I think I was the person who spent most of the time with you in that lab from 1994 to 2006. So almost 12 years. And I learned a lot from you. And when I was there, I remember Eric, Craig, Bart, and Austin. I remember Karthik, and so many of you were there. And it was a very, and Jeannie, and then Heidi. It was very nice when I entered the lab. I felt very happy about it. And then I learned a lot from Mark. Mark was so patient enough to, even if we do not know anything, he will be like a real teacher. And then it was a very great experience. And then G was there, Gina. I remember everything. Those days we can never get back.
Right.
Very wonderful time we had. And then I entered a very nice lab with Dr. Mark and Cindy, I remember. And then Mark's sons, I remember they had small pet. That pet also, I remember when I went to your home.
Oh.
Remember that small?
The pet?
Was it a snail?
Oh.
No.
Bearded dragon?
Draco, the bearded dragon?
Yeah. It was real fun. And then that time was very wonderful, and cannot have that time anymore. But we have to keep that in our memories. And it was nice. And like Karthik said, everything, whatever I learned after, initially I was only just even crystallization, maybe crystallization I did, and then protein, and then crystal structure. I started with small molecule crystal structure in India, and then I came here and then started working on proteins, but I never went to protein preparation and then those other things which go before the crystal structure. So all those things in Mark's lab I learned, and then I enjoyed the whole life there very much. I still remember, and then wish you a very happy and long and wonderful retired life, Mark. And then
what are you doing now? Are you retired now?
Yeah, I spent only 2 years after your lab in Ron's lab.
Yeah.
And then I due to some reason, because my son wanted to go back to India.
Yeah.
For good. So we sold everything, all our house, car, everything we sold. And then within 8 months, we came back here. And then, but I still, now what I'm doing is I'm still continuing teaching music to the children here. And then they are performing outside, and I enjoy life here, but still I remember the crystallography experience and things I got from your lab. My husband is there. I will call him a little bit later.
Okay. Well, thanks. Thanks. We the lab really appreciated all your contributions. So many different structures.
Everything is only with our colleagues, and then your only with your help I could develop so much in the lab. And I
Right.
still remember Craig, Eric, we all had good time.
Yeah.
And when I entered, they were all there. And then later on, Bart was there. Bart was there. Everybody, it's so nice to see everybody, Karthik and I remember Austin. Austin, do you remember me?
Okay, let's move on. I think Sarah is next.
Okay,
Thank you, Viji.
thank you.
Sarah?
Am I the only Sarah here?
Yeah.
Okay, so I was part of Mark's group from 2012 to 2013. So Mark's group was the first wet lab I ever worked in. So Mark was gracious enough to take my very inexperienced 17-year-old self. I was just a kid back then and I knew nothing. I was just very excited to do research, very excited to study biophysics, and that was what I ended up majoring in. And I'm just very glad that I got that opportunity and was able to learn so much in the lab in terms of biochemical techniques. And my research interests ended up evolving over time. And right now I'm actually working on stem cell differentiation.
Wow.
And I'm doing my PhD in a biomedical engineering department. But a lot of the techniques that I learned working in the lab, I still use today. And it's crazy how many years it's been since then. But I also want to thank Matt and Kathleen for teaching me so much when I was in the lab as well. You know, from running gels to, you know, how to not break the ultracentrifuge. So all of this has stayed with me. So I'm so thankful that I got that first opportunity, and I want to wish you a very happy retirement, Mark.
Thank you very much, Sarah. Yeah, good luck to you. And as you, have you finished graduate school or you're still
So I'm a second-year graduate student now, but I'm in the 5th year of my degree overall. So I still have
it's an MD.
About 4 to 5 years left.
Okay, it's an MD PhD?
Yeah.
Oh, wonderful. Impressive. She's in Wisconsin. Yeah. Okay, let me see. Next is Eric Fowman.
Hello, good to see everybody. So I think I was in the lab from 93 to 98, 5.5 years, and so overlapped with a lot of you guys. Let's see. When I joined the lab, I had 0 children. When I left, I had 1, and now I have 5.
Wow.
All boys. Youngest is 10. If I can tell from the camera mark, it looks like that's the room where Noam's bris was. Is that? Am I remembering correctly? No.
Different
No. Okay.
Different house.
Different house. Oh, okay. You moved. Okay. Well,
Oh yeah. Right. Yeah.
Well, so we of course moved in 2000. So when I left the lab, I went to Park Davis, and in 2000, Park Davis was bought by Pfizer. And in 2007, Pfizer decided they didn't need to be in Ann Arbor anymore, and so we moved to Cambridge, Boston, Newton. And so I've been with Pfizer ever since. So this is my 23rd year now with
Wow.
Pfizer. I have a group of 10 computational biologists, and we do genetics and bioinformatics and image analysis within our internal medicine research group. But it's an exciting time for structural biology, frankly. One of the resources we have now is something called the UK Biobank. So they've done whole exome sequencing in 500,000 people. So we have the exact amino acid sequence of every protein of every one of those 500,000 individuals.
Wow.
And one of the amazing things that we're discovering now is that when you get that large number of people, almost every amino acid in every person, almost every amino acid in every protein has been changed in at least one person.
Wow.
So all the same information that we use and think about, and the mutagenesis that we used to do, you basically have the thought experiment of, well, what if we change this amino acid to something, put it in a person, and come back 60 years later and see what's happened to that person. So it's all the same thinking, and all the structural biology, all the thinking about what these amino acids and proteins are doing, we use every day.
Wow, that's great, Eric.
I enjoyed my time there. So Mark, when's your, I don't know, last day? Is there such a thing?
Last day was a week and a half ago.
Ah, okay.
Yeah, so I have to admit that cleaning out everything was quite, had goods and bads. It was a lot of, very emotional to go through everything. But I successfully went through everything, and now there'll be a new people moving into my space, but I'm still affiliated with the department, so we'll see.
So was your lab space still the lab in the chemistry building?
Well, I left the chemistry building in 2015, right before my sabbatical, where I went to work with Suzanne Buchanan at the NIH. And then when I came back from the NIH, I got space in the biochemistry department in the medical school. So I had about 3 days there, and I think the people who joined me there will remember that. And then I moved across the hall to another lab, which was smaller, so I was able to throw out more things, and now finally throw out everything else. So except a lot of other colleagues took a lot of the stuff. There was very little that wasn't used up, but yeah, I really I'll probably miss definitely looking at protein structures and tracing chains and looking at maps. Was the funnest part. And Eric accomplished so much while he was with us, and I'll never forget your contributions, and I appreciate them, and I'm so happy that you now have a bigger group than mine.
I really enjoyed the time.
Thanks, Eric. Craig, you're muted.
Let me unmute myself. Hi, so those who don't know me, I'm Craig Smith, and so I was in Mark's lab from 93 to 2001, just got in there in January. So yeah, so I overlap with a lot of people here I see. So people I haven't seen since I left the lab. So it's good to see you guys again. So yeah, when I came to Michigan, I went to undergrad at Purdue University, and I was fortunate enough to take a crystallography class. And when I took that class, I fell in love with that stuff. I couldn't wait to solve my first structure. But all I had was theory. So it was great when I came to Michigan, and I did a rotation in Mark's lab, and I realized this is what I wanted to do. And I'm so thankful that he gave me the opportunity experience. Up to that point, I don't think I had purified a protein. I think I did my protein purification and crystallization stuff all sort of when I was at in your lab. It was great to have people who are already there. G was already there, and Tom and Heidi, and so it was great for them to show me the ropes in the beginning there. So I'm thankful for all you guys, and thankful for you, Mark. You were, you taught me a lot. I learned so much, and so after I left Michigan, I went to St. Louis for my postdoc. And I thought, I want to stay 5 years, and here I am 20 years later, still in St. Louis at Washington University. I teach in the biology department. So I actually teach a class in structural bioinformatics.
That's great.
That I created myself. And I started that class about 4 years ago. And actually I collaborate with Bart on he actually supplies me with some of the protein structures we use in the class. And so, I use my connections I got here working with you. I still have them.
Right.
So I see Bart every other week, except I did miss this Friday. I apologize for that. Yeah. So like I said, I teach in the biology department. I teach this bioinformatics class. I also teach introductory biology. I still try to do some. I don't really do. I try to do some. I got a paper out this past year and trying to get more out. So still trying to do it. Don't have a group. I have a group of undergrads who help me when they have time, but I don't have like an official lab or anything. But it's been fun. It's been great. being able to take the knowledge I learned from you and pass it forward to the younger students. So congratulations, Mark, on retirement. And
miss you.
Yeah, no, I
You
missed
were
you
a
too.
great
And yeah,
lab, and I liked how you visited occasionally afterwards. And
yes.
And I have some pictures that you're in later. I'll show you. Get that part.
Also, I don't see Ellie, but Daniel Noam too. I remember when Noam was born, and he's like a PhD chemist, I think, last time I heard.
Okay.
Big deal now somewhere. So that's awesome.
Okay. Yeah, Noam, do you want to say hi? And then Daniel? Noam?
Yeah. Hi, everyone. I think many of you know me. I'm Noam, although I mostly just remember people's names, unfortunately. I've been in the Saper lab since '93 to probably last time was 2015. I did my undergraduate degree in chemistry at Brandeis University from 2011 to 2015, doing mostly organometallic chemistry, and then went on to do my PhD at UC Berkeley with Professor John Hartwig in organometallics, basically studying the mechanism of nickel catalyzed cross-coupling reactions, specifically with N-heterocyclic carbenes as ligands. So really diving into organometallic chemistry. And now I'm currently living in New Jersey with my lovely wife, Rachel. And so we have a job, I have a job as a senior chemist at a small pharmaceutical company called Exemplified Biopharma. I'm doing mostly process chemistry and bringing molecules to market.
Great.
Rachel.
Yeah, thank you, Noam. Hi, Rachel.
Hi.
Glad you guys could join us. Daniel, you have anything to say?
Yeah, we're so happy to be here.
Sure. Can people hear me?
No, I don't think they can. You better unmute.
Come here.
Yeah.
We can hear you, we just can't see it.
Okay, cool. Hi, everyone. I'm Daniel. Similar to Noam, I mostly remember names, not faces. Let's see. I didn't follow the science path. Instead, I'm a computer scientist. I work at Autodesk, which is CAD and CAM software company. So I've been working there for about almost 6 years now. Yeah, it's great to see that everyone showed up though. Thank you.
Thanks, Daniel. That's great.
Ellie, I'll tell what Ellie's doing.
Oh, yeah.
So
Briefly.
Ellie called us just before this to say hello to everybody. Ellie lives in Israel, so he was going to sleep when he called. And Ellie has a wonderful wife, Abby, and a daughter who's 5 and twin boys who are about 17 months old. So they're pretty busy. And he's a farmer.
Yeah.
So although he did get his degree in biology.
Yeah.
All right.
Good. So
Matt.
we're going to continue with Matt. Matt.
Hi, Mark. So I was up in the lab in, I think, 2012 to 2013. And I'd come out after a PhD in crystallography and then joined Mark's lab to explore the world of membrane protein crystallography. And so that was an exciting adventure for those years. And I have really great memories of both the time in the lab and also just Ann Arbor as well too. And I definitely think back about the times we went to Zingerman's and all these different places I love. Think about the river and just the school. So it was a good time period for us. When I was up there, I think, Mark, you met my wife, Epi, and now we have 2 kids. So we have a 2-year-old and a 5-year-old. And it's been an exciting adventure to kind of get into that realm of parenting with them. We managed to get back to Wisconsin. So now I'm in Madison. And I joined up here in about last January, and there's a start of the Cryo-EM Research Center. And so I've switched over now to that side of things of trying to get into cryo-EM, single particle analysis, and also there's a big tomography side of it.
Right.
Some of that stuff is really pretty exciting and fun. People are trying to solve structures from inside of cells with tomography. And just there's a lot of excitement happening there. And we're trying to build out a big center that's both with the University of Wisconsin, also 4 other universities that are building big national centers for people to submit studies for tomography.
Yeah. Well, definitely cryo-EM is giving the crystallographers a run for their money. Though some of them tell me that interpreting 4 angstrom maps is quite challenging sometimes.
Oh yeah, I'm sure. And I think the maps are different. You know, they
Yeah.
definitely look different than the crystallography maps. In
Yeah.
some ways they can be clear. In some ways they're just, the side chains are different.
Yeah.
But we got to, I think, 1.8 angstrom already here
Okay.
on our instrument. And yeah, it's definitely an exciting time. The technology is advancing very quickly.
Mhm. Great. Thanks, Matt. Kathleen, you're next. You want to unmute?
Yep. I am Kathleen, and I was in the lab with, well, from I think the end of 2011 to 2014, and I overlapped with Matt and Sarah. But when I was in Ari's lab, I did alkaline phosphatase crystals with Eric, and then also our lab moved over. So I worked a little bit with VG. I mean, we were in the same space, so I'm not sure if you remember me.
Yeah.
Right now I'm with Sarah Veach, so I'm in Martha Ludwig's old space, and I'm set to retire. I keep pushing it off, but I'm not September 1st this year, so it's right around the corner.
Yeah, yeah.
But
Kathleen,
it was a, oh, go ahead.
Real boost to the lab. Really.
Pardon me?
Really
Oh,
helped. You did so many techniques and things. It was great, great.
A lot of things, boxes.
Yeah.
Yeah,
in
we
the
have a lot of
basement.
boxes
In the basement.
with
in
your
our
name on
basement.
it. Yeah.
Yikes.
So what are you
Yeah,
doing
so-
now? Are you doing any specific type of research or you're?
Well, with Sarah, we're doing the single molecule, the more like, now I'm forgetting the acronym, but we look at photoactivatable probes.
Oh.
So that's what we put in. So we look at where proteins localize in the membrane, inside, outside, if they're in the liquid or the gel phase. So I do more of I take care of the cell line. So we do transfect these cells into about 10 different cell lines. I mean, we have many more than that. And I don't do the imaging. So I do much more of taking care of the cells, the molecular biology. But even that, I'm kind of handing off
Good work.
as of getting ready to retire. I actually only work 3 days a week also. So life's pretty good right now. But yeah, it was fun working with Matt and with Sarah. And I actually work for the department a little bit too. So yeah. But it's exciting. So yeah, pretty excited for you and Cindy, and traveling and doing things.
Thank
It'll
you.
be fun.
It's good. So you'll have fun after you retire.
Yeah,
Becky-
well,
Yeah, thank you, Kathleen. Becky?
oh, you're welcome.
Hi, everyone. I'm Becky. I feel I'm a footnote of a footnote. I was just looking for a job as an undergraduate in 1996 or 7. And I went to Mark. My father is a scientist, and he said I should knock on Mark's door. And so my main job was to wash dishes. But also Eric and Bart and Craig let me crystallize proteins. So I crystallized CDC25, which is, you will all know what that means. I still don't know what that means. I majored in arts and ideas at the residential college. And I work in the arts, creativity, entrepreneurship now. But I've been sitting here thinking of, there's 7 things I think I learned from you, Mark, in those sort of 3 years together. And you've all referred to techniques. And I think for me, techniques, they weren't about science, although they were, but the first one was about in comedy, there's this idea of you yes and people, and you build on their ideas. And when I knocked on Mark's door, even though I said, I know how to crystallize proteins, that's about it. But you yes anded me, and you said, okay, let's do this, figure out how you can help Eric and Bart and Craig and VG in the lab. And then the second was about the patience of all of you guys, because I was just an undergraduate finding my way, and you were so patient. Also, I guess, waiting for the crystals to grow, but actually just patient with me and trying to learn things. The third was, which I've tried to do with the teams that I lead, is helping people figure out what they're good at and encouraging them. And, we're not all good at everything. And so how you help form a perfect team. The fourth is, I'm now hearing Eric speak again and Craig again, is how you excite people about things. And Eric, I'm just so excited when you talk, even though I don't work in your field. You're so inspiring. And Craig, I want to take your course, even though I would fail it. But I'm really inspired by you guys. the fifth, and now because I know about Noam and Daniel, is the sanctuary of the lab. And I have two kids now, an 8-year-old and a 4-year-old.
Oh.
And that cold room, I now know why my dad was always going back to the lab with 4 kids. That cold room was such a sanctuary from home life. The sixth is experimentation, and just the magic of experimentation. So I worked just recently for the British government for about 3.5 years, leading a global experiment for them in creative entrepreneurship and developing economies. And how you get a group of people to experiment and be excited is what science is. And then the final one is about collaboration. And a lot of my work now is thinking about ego in collaboration and how you solve a problem. I'm working on economic unemployment using creativity and entrepreneurship, but how you solve a problem together when you have egos. So all of that, I think, Mark, you created a spirit, which I can see still here to this day, in that space that meant a lot to me. And then I finally wanted to show you that I have my artwork on the wall there. I don't know if you can see it. I asked my father to remind me what it is. I'll tell you what it is, because you'll all know what it means. My father, it was his first electron density map of profilin actin at 6 angstroms resolution, but none of my guests need ever ask what it is. They think it's a beautiful painting to me, but you'll understand what it is. So thank you for inviting me. I feel really touched to be here. And thank you for all of you guys. What you taught me really has stayed with me all these years. So thank you.
We loved having you. Thank you for those kind comments. I know you were a real contributor to several of the projects, and
I don't think I was, Mark. That's very nice of you to say.
Eric spoke very highly of you, definitely.
Well, thank you, Eric.
Yeah, we loved having you there. Okay, thank you, Becky. Meredith?
Hi. So I was there as an undergrad, so I was also learning quite a bit. I'd never had purified a protein on my own, and I got to learn how to do that. I was there from, I think, 2016 to 2018.
Right.
I can definitely thank working in the lab and Mark for passing like all my biochem classes. It's just like doing it in person made it make so much more sense than, learning it in a lecture. And then I'm in medical school now, so I'm starting my 4th year. Started it 3 weeks ago, and we'll be applying to residency this fall. So
great.
Yeah.
That's wonderful. It was great
What?
seeing you, Meredith.
What residency are you looking for?
I want to do urologic surgery, so like female pelvic medicine, reconstructive surgery. So I've given up the bench research, but I still get to do some like clinical work, which is really fun.
Good, great. Thanks, Meredith.
Thank you for
Austin,
inviting me. Congrats.
Yeah. Austin, I think you're up next.
My turn? All right. Hey, yeah, so I've been, let's see, I was in Mark's lab 2003, I think, to 2007. And I came there as an undergrad. I just needed an independent study, and I just kind of sent Mark an email, and he let me come work in his lab. And that's where I learned a whole bunch of stuff. And I stayed there as a tech afterwards for a couple of years, and then I ended up in California for a couple of years. So I worked for a biotech company there and decided to go to grad school. So I decided to kind of change gears. I did biomedical engineering at UC San Diego, and now I'm in Virginia Beach, and I've been in Virginia Beach for like 9 years, and I work at a company, we make tissue grafts. So we make some regenerative medicine products, which is very cool. So I get to do some tissue engineering and a lot of that. 3D cell culture, some stem cell stuff, and I actually got the job there because they needed someone who knew how to use an HPLC and purify proteins. And which was funny because that's not what I really intended to go for again, but that's how I ended up there. But I got to do a whole bunch of different and exciting stuff since I've been there. So I'll be here for a while, I think. And I definitely remember VG and Karthik and Katie. And she just showed up. And yeah, congratulations, Mark. And I was happy to see that something I worked on there ended up on a paper eventually.
Absolutely. Yep. It was great.
It was very exciting to see. So happy for you, Mark. And
thanks a lot.
Enjoy yourself.
all the best to you. That was great.
Thank you.
Thanks, Tom. Austin, we'll move to Tom. Tom Transon.
Okay. Hi, everybody. So I'm going to say a few words about, first of all, Mark, when I got the email that said you're retiring after 30 years, I said it can't be 30 years because I finished like 25 years ago and that doesn't add up. And I remembered that I didn't finish when you started. So Mark and I started about the same time, around 1990. So I'll say a couple words about what I've been doing, then I'm going to see if I can embarrass most of the Sapers on the call here, but we'll see. So I started in the lab. In those days, the biochem department did 3 one-month rotations. So I did a rotation with Charles Williams and then with Vince Massey. And I wouldn't say that they were way too intimidating, but they were probably some stratospheric level of intensity. Then I got in the lab with Mark and it was relaxed and fun. And I got some crystals after 1 month. Now I didn't purify the protein, so that made it easier. So that just looked like it was the thing to do. And I love the idea of crystallography because it had this math involved in it, and I've always loved math. So yeah, I got going. I was the black sheep because I was working on a boring lectin, and everybody was doing these protein tyrosine phosphatases and everything. And I got to go to these seminars and hear all this stuff, and then I would say, yeah, I've got a protein that binds sugar. But it was fun. It was a great thesis project. You know, I don't know if people are publishing Nobel Prize work based on my paper or not. I haven't.
I've actually seen a lot of papers on amaranthus seed protein, so
Well,
it's still out there.
It's still out there. That's right.
Yeah, they're referencing your paper, so
yeah,
that's good.
So the other thing I was going to say is that in some ways my career has gone like academically, like stepwise down in terms of I'd say rigor and that really important focus on the key details. So after my PhD, I finished in '95 and I went to Belgium and did a postdoc at the Free University of Brussels. Met some really great people. I didn't have to I didn't have to purify protein again. In fact, some other people were crystallizing it. So I solved a couple of structures, most notably a camel antibody, which was a single domain, heavy chain only, antibody naturally occurring.
Which are real big deals now.
It was a big deal.
Very important.
Yeah. And I kind of lucked into it. I was third author on the paper and that was fine. It was a great paper.
Mm-hmm.
Then finished that postdoc about early 99 and came back to the US and decided to work at NIEHS, the National Institute of Environmental Health Sciences, which was a change because government labs, they just have money. They don't have to write grants. And I think that unfortunately makes them a little bit less hungry for like the really, really fine detail. I think that some of the scientists are kind of like, just go to work and get the work done. I had a good experience, although I did crystallize and partially solve the structure of the wrong protein.
Yep, it happens.
So I was trying to crystallize an RNase, and I got to MAD maps, and the seleniums were in the wrong place. And so after a bit more work, I determined that I had crystallized and partially solved carbonic anhydrase, which the bacteria grow because you're forcing them to grow in this high CO2 environment. So all of my protein had precipitated out and there was nothing but carbonic anhydrase left. And the molecular weight was 900 Daltons different, so it looked almost the same. And it co-purified through nickel ion exchange and gel filtration. So crazy thing. I think it started telling me that crystallography wasn't for me. So I did a postdoc in a little bit more theoretical work with Tom Darden, whose big thing was some polarizable force fields that he still worked on a bit. And then didn't have a job for 7 months, and that was a little bit weird. And then I took a job at the EPA because it turns out that other kinds of scientists sometimes are not good computer scientists, and they need people to fill in and write software and figure out what they're doing.
Mhm.
So I've been at the EPA as a contractor. Basically, every EPA scientist, when they need help with their data and their science, come to the group that I work in. It's pretty low stress. It's been great.
Great.
So I have two kids now, 16 and 13. But now I have to tell a couple stories about the lab. So the first one was, you know, Mark and I were somewhat alone for a year or so. And then as people started coming in, it was more stressful. Mark had to be at the lab a lot. And, you know, when you're at the lab and you don't have a chance to eat, sometimes you got to eat some like cookies. And sometimes like the only cookies around are your grad students' cookies. So I come in and my cookies are gone. And Mark said, I'll get you more. Sorry, I'm sorry I ate your cookies. I'll get you more. But then he didn't get more because he said, if I get more, then I'll eat them. Right? He didn't want to be tempted.
I don't remember that, but thank
And then
you.
it turned, you don't remember that? So then it turned out one time I had Oreos, but he didn't eat Oreos because they had lard in them.
Back
so it turned
then
out
they
Oreos are the solution to cookies that don't get stolen. So the next thing is that one night I was working and I sent Mark an email. It was like 11:30 or midnight, and he wrote an email back. And then I wrote another email, and then he said, let's get on chat. So we started talking back and forth. And then about 1 a.m, he said, I have to go because my wife thinks I married a computer. So Cindy was very patient.
Very patient.
Still is, I'm sure. All right, so the next story was, so Noam was born on Shabbat, as some of you may know. And 1 week after his birth, there was a celebration at the Saper's house. And it was really hot. I don't remember, is it June or July?
August.
Okay. So it was really hot, and the whole house full of people, many of them very observant, but Daniel decided to unplug the air conditioner. So after about 20 minutes, the room started getting kind of warm. And Mark's mother said, nobody's touching that plug. Someone said, let's just plug the air conditioner in. And she said, don't touch it.
Mark's mother.
So thankfully, Mark knew that there was a goy around who didn't mind helping out. And I plugged in the air conditioner. Everybody cheered.
Yeah.
That
You
was-
remember that.
I don't remember
great.
that at all.
A great memory.
Great.
Okay.
So, Mark, you know, it was a great time, and clearly you have a lot of great students you've brought along, postdocs. So, thank you.
Yeah, thank you, Tom. Great memories. I remember when we solved the non-crystallographic symmetry of your crystal. That was a real fun. It was a real big deal, and you got to use some of your math. So
Some really basic geometry, but applied to real science. So it's great.
Okay, thanks, Tom. We're going to move on. We're going to make it through this list. Bart.
All right, hello. So I was in Mark's lab the 94 to 2000. I had during that time 3 kids in Mark's lab. So I really appreciate Mark's patience with me as we went through that process. And every now and then I would take a month off to have a child. But yeah, so that was great. It's really great to see all the faces and old friends. So thank you all very much. During, you know, as with most of you, you know, we learned purification and crystallization and crystallography. I grew a love for structural biology, so I'm still solving structures. I've got one right there that I'm working on. So I love solving structures. I went after Mark's lab. I worked at Emerald Biostructures, which some of you may know makes the Wizard crystal screen. I think it's made by Rigaku now because it's been sold, but I worked there for a number of years, and we started a business solving structures for hire for drug companies and stuff, and we did that for quite a while. I have sort of come full circle. Now I'm back at Seattle Children's Research Institute. I work for the Seattle Structural Genomics Center, which solves structures. So that's why I like to work there. We've solved, hundreds of structures, and we started working with Craig a few years ago, helping bring in structures. We don't write a whole bunch of papers, so it's really great. I always love solving structures and hate writing papers, so I get to keep doing that. And then Craig's students have sort of contributed writing some of the papers of some of the structures that we have solved.
Great
That's
collaboration. That sounds good.
Yeah, so we've been working on that for probably 3 or 4 years now, where Craig and I have been talking. He came up to Seattle and presented, and it was pretty fun. I did my first cryo-EM structure recently. It was about 3.1 structures. So we've been doing some cryo-EM. And I ended up I never always wanted to go into industry, but have come around to working in an academic lab, and I actually got an R01 funded like last year. So
wow, fantastic.
Pretty happy with that. And it's a drug discovery type program, so we'll see where that goes. Yeah, it's so great to see your guys' faces, and I really do want to thank Mark for all the patience and training over the years. And thanks a lot.
You've been in Seattle this whole time.
That was great.
Pretty much, I guess I came from Seattle, wanted to move to Ann Arbor, and then I moved back. I was looking for that job when I moved back because my mom was sick, and then I took up with Emerald and just have stayed on. So yeah,
nice, great to see you.
Fantastic. Okay, Bart. Thank you, Brian. I'm glad you could join us, Brian.
Thank you. Sorry about that. Well, Mark, thank you for inviting me. Some of you know, especially like BG, Craig, and Bart, I was in Mark's lab from '99 to 2003. And I struggled a bit with my research, but one thing I really did get appreciation of is troubleshooting, and it comes into my career later on. I also learned that the right path for my life may not be the most obvious one. But I was still happy and glad to support the research in Mark's lab, and was happy to see that AGT paper was published last year. So, if you're ever worried about something, just wait 15 years, something will get published.
Yeah. That was one of these.
I left my lab. I left the lab with a master's, and I proceeded to get 2 more masters. So I feel like I should trade it in for a PhD now. So I got an MBA and a master's degree in industrial and systems engineering. That led me to, I worked over 15 years at Thermo Fisher Scientific in Illinois in various roles. Actually, in January 2020, I joined ABCAM, which was based in Cambridge, Massachusetts. I moved out to Massachusetts in April, right at the beginning of COVID. So that was extremely fun. I am currently the head of operational excellence at ABCAM, which means I help to improve processes and various things like that. Our company actually just moved to our new site in Waltham, so not too far down the road from Brandeis. Actually, it's probably within a few miles. And I get a chance to use my business and engineering skills, helping to solve issues with the production of antibodies. So combined everything all together. And just would like to say that I'm glad to have known Mark and Cindy and the family. Looking back, I was very thankful of the kindness that you shown me when I was at Michigan. One of the things that I did remember from back then was I was very honored. I forgot which son, but I was able to attend his bar mitzvah. So that was quite an honor. And I thank you for that. I wish you best on your retirement. And if you're ever in Massachusetts, you're welcome to visit.
Great. Thank you, Brian. I'm glad you found a position that really is working for you. Good.
Thank you.
Good.
it's good to know that, yes, the wait 15 years and the
Wait
paper may
15
still
years.
come out.
Yeah, yeah. Okay, Katie?
Hi, everyone. Mark, thanks for inviting me. It's been a long time. That was kind of a nice trip down memory lane. Congratulations. I was in the lab as an undergrad from, I guess, 2005 to early 2007. And let's see. And then after that, I went to medical school and residency and then fellowship. And now I am a GYN cancer surgeon, oncologist at the University of Chicago. So I do mostly clinical medicine. I do a little bit of research, mostly more on the clinical side. But when I was in the lab, when I was in your lab, I did a lot of Western blots and troubleshooting how to do that. And that later served me well in my fellowship, because then I had to do that again. And so I was able to hit the ground running a little bit. But I think mostly what I learned was just kind of problem solving and I don't know, just kind of figuring out, like being curious and thinking about what the next step would be, and kind of taking something through a journey of scientific exploration, I guess. So I had a wonderful time in your lab. I'm grateful for all the experiences, and congratulations on the next exciting part.
Katie, thank you very much. Good luck in your position now. Really, I'm glad you could join us. Fantastic. And Aaron.
Hey. Hey, yeah, long time no see. Although not too long, because
no, you're the shortest.
Right, exactly.
Most recent.
Yeah, no, I was there from as an undergrad from 2015 until about, I think we went into 2019, right?
Yep.
And yeah, I didn't overlap with many people, but Meredith, I overlapped with you, and then I think I met Kathleen there at the beginning too. We had like maybe a year where we saw each other. But yeah, that was I started like the second semester my freshman year, so that was really like the my first experience with science, like really doing research. And yeah, I'm thankful to Mark for many reasons. He was very patient with me as I like precipitated protein in the FPLC various times probably, and I think I spilled my purified protein a few times too. But yeah, it was a great experience, because it was a very independent research. I think it was very unique, and Mark was always there for me, so it was really great. But yeah, then I've kind of changed gears a little bit. So I graduated in 2019, and then I did like a relatively short internship, about like 4 or 5 months at a neurotech place. And so I kind of started doing a little bit more neuroscience, and then I decided I'm actually in Mexico now. So I went to Mexico, study. I got some like funding to go to a lab and do some computational neuroscience. And I did that for a year. And then right to the institute, there was a mathematical institute, and I had like studied some math in undergrad. So I decided to do a master's in math. So now I'm here 1 year into my master's in math in Mexico. So yeah, I deviated quite a bit, but I might head back to structural biology. I mean, I definitely have some really great experience, so I might go do that. But yeah.
Great. Go into cryo-EM. That's where the action's happening.
Sounds like it, yeah.
Yeah. Great, Aaron. Yeah, Aaron was a sole member of the lab for quite a while, but contributed on a lot of projects. I think we got a couple of papers out, so that was really nice.
Yeah. Some crystallization. We solved that first structure was really exciting, because we were like going through it. We were remotely, shooting the X-rays, and there were always like 5 or 6 angstroms. Then we got like one that was like about like 2.1 or something. I can't remember. And that was exciting for me. Yeah. That's cool.
Okay, thanks, Aaron. Thanks for joining us from Mexico.
Yeah, thank you.
Spanish must be a lot better these days.
Oh yeah, it's definitely improved a lot. I don't know if that means it's good, but I've definitely improved the level of my communication. So
great. Brahmra.
Congrats.
Yeah, thanks, Aaron. Brahmra, so happy you're here.
Hi, Mark. Hi, everyone. Sorry, I'm traveling, but I wanted to make sure that I wanted to wish you congratulations on the retirement and thank you. I think, so after I was in your lab for about 2 years, 2007, 6, 7 to 2008 or so. that was the bridge to move out of academia into commercialization, business consulting. And now I'm working in medical devices. We make cochlear implants. I enjoy it. It's been a wonderful journey. So what stood out to me was you were one of the most humblest and kindest boss that I ever had in my entire life.
Thank you.
And that stayed with me. It was a very safe space, and I was able to explore with you what I wanted to do in my career, and you were very supportive throughout. So thank you.
Thank
And
you.
congratulations.
Yeah, really appreciate it. Great. Well, I think we went through everyone. I don't think I missed anyone. I have been in touch. Tom, I wanted to mention that I was in touch with Alex Smith. I guess he couldn't make it, but he's doing really well. I constantly see new publications from him. He's an MD, Masters in Public Health, and has his own operation. So I've seen him, and I got a lot of.
I emailed with him a couple of times. I looked him up just for some reason, and he's in California, right? Is he in California?
Yeah, California, definitely.
A couple of kids and doing really well.
Yeah. Well, to continue the events, I appreciate everyone hanging on. My son and wife put together some stuff that they wanted to show. I need to give you.
Do you want to show the photos first? Or you want to
No, Daniel wanted to show.
I'll do it. So
wait, should I share your screen?
you can give me here. I can.
Co-host.
You have to. Yeah.
Okay.
Give me co-host.
Okay, good.
All right. So I've got a very small PowerPoint or I guess whatever the called keynote.
You're muted. Oh, you know that.
Yeah, I don't want feedback.
Okay.
You can hear me though.
Yeah.
Yeah, we can hear you.
So I've got a small PowerPoint. We've got some surprises for Mark, and then Mark has a bunch of photos. I think,
well, let's see your photos
All
first.
right, so go ahead and share the screen here, and I will go ahead and play the slideshow. So welcome everyone to Mark's retirement party. Congratulations, Mark.
Thank you, Daniel.
We have this wonderful picture of Mark. I don't know where this was taken.
That was at a synchrotron.
Okay, well,
I think
a lot of these pictures I just found online. Googling Mark. So with 30 years of the Saper Lab, 1990, 2021.
Right.
So Mazel Tov.
Thank you.
We have, this is from the University of Michigan website, I believe,
right.
Of Mark.
Yeah.
We have a picture from outside the lab. I couldn't find one inside the lab on Google, but this is a very nice one. We've got pictures of the Saper family here. So we have, this was, I believe, at Noam's bar mitzvah. We have some of the family over.
Explain that's Ellie and Abby.
This up in the upper right, we have Ellie and Abby and the rest of the family in Israel. They're on the left there. And then in the bottom, we have Noam and Mark. This is probably at Brandeis.
Brandeis, yes.
So that's very exciting. We have another picture here of, I don't think this is Ellie, but this is young Mark. Younger Mark.
Not much gray hair.
Yeah, there you go. I like the smile, which is why I included it.
Okay.
Good smile. We have this, which I believe this is at Noam.
This is at Brandeis.
Is this at Brandeis?
Yeah, Brandeis.
No, this is my undergrad. This is when I graduated. I showed them the lab at Brandeis.
Yeah.
But Noam, didn't we get in? You got a little trouble that.
Yeah, I was just wearing sandals in lab because I just came in to show him. But
yeah, no. I included this picture because I thought it was awesome that Noam followed Mark in science. I didn't, but so I have this picture of the Pew Scholars came up when I Googled Mark, 1983. It's a very good picture. I like it.
It's a funny picture.
Yeah,
that's a funny picture.
There was I looked up the Saper Lab papers. There's 30 plus papers just with Mark's name on it. It's 30 plus I think. This is just the lower bound. Was there artifacting? Okay. Bacteria that you guys looked at. There was Yersinia. I'm gonna mispronounce these names because I'm not a scientist. Yersinia enterocolitica, Haemophilus influenza, and Escherichia coli, and there were many others on the list, but these are the ones I picked. We've got some protein structures that Mark and the rest of the lab solved. I asked Mark to just pick a couple. This is not an exhaustive list. We have 1C25, the human CDC 25A catalytic domain. Yeah. We've got,
and these pictures are all made by Daniel. So
with Mark's help,
we got a new structural biologist.
Yeah, right. We have 1VHR, the human VHI related dual specificity phosphatase. We've got 1YPT, the crystal structure of Yersinia protein tyrosine phosphatate at 2.5 angstroms in complex with tungstate.
Excellent.
We've got 3CKM, LPOA, YRAM, C domain from Haemophilus influenza, a regulator of PBPIA. 1A, sure. And we have 3P42, structure of GFCC YMCB protein encoded by the E. coli group 4 capsule operon. And finally, 6U2A, Shy-A endopeptidase from Vibrio chloria, the open form.
Yes.
And lastly, a shout out to Pymol, which I used to generate these images. Thank you, Mark, for helping me. It is the software that when I would walk into Mark's office, I would always see him using.
Yeah.
And so, very exciting. All right, so that's all of the presentation, very short. But now we've got some surprises for Mark, so I'll go ahead and start us off.
Okay, go ahead.
So I have a gift for Mark.
You do?
Yes. So Mark, you can go
Wow.
ahead and open this.
I got a big box, a little box, but very heavy.
Be careful with it.
I will. Oh, wow. Whew.
You can show it to the camera. I don't know how we'll be able to see it.
It's one of these crystals where they use lasers to make a crystal structure inside, and I think it's 1Y. Which one is that?
It says it on
It's
there.
the 1YPT. I think that's Yersinia.
Yes, Yersinia phosphatase with tungstate, right?
That's with tungstate. Is that a dimer or is that what it looks like?
It's both of the.
It's both of the molecules in the dimer.
G's protein, right? Didn't G do that?
What?
Didn't G work on that?
No, it's Heidi.
This was Heidi and Eric worked on this one with tungstate. Yeah, it's great.
So
thank you.
Yeah,
It's
I
beautiful.
like the crystal. You know, it's a crystal and a crystal. Or you know, at least
That's
kind of,
crystal and a crystal. Thank you very much, Daniel. That's really sweet.
Yeah, you're welcome.
Thank you. Wow.
You don't need that. I'll type later if you don't want it.
Thank you very much. I wasn't expecting this. This is indeed. And then I think Cindy, what's up? You have something?
She's coming. Hold on.
Well, this has really been a special time.
Here, we have a cake for you.
A cake?
And you'll have it later. That's okay. And we have another picture for you, another present for you.
Another picture. Okay.
You can blow out your
Okay.
candle if you want. For many, many more years of
Thank you,
successful
Cindy.
Saper Lab.
Wow.
Is that an M for Michigan or for Mark?
Either one.
Yeah.
Either one.
Or a Sigma?
So Mark, we knew that you might not know what to do with yourself if you're not solving protein structures. So maybe this will help you a little. You can solve something else.
A lot of wrapping.
Sorry.
Oh, wow. It's a puzzle. Oh. It's a puzzle, and I think it has all sorts of things from Michigan, including protein structures and pictures of people. And it's very neat. Thank you.
500 pieces.
500 pieces.
Yes.
Thank you very much, Cindy. That's really sweet.
For Daniel.
Yeah, thank you, guys.
My
Thank you.
catfish skills being as they
Yeah.
are. But
And beautiful pictures that Daniel made.
Yeah.
Congratulations, Mark, 30 plus years at U of M. How sweet.
and the
Thanks.
Saper Lab.
Yeah, thank you.
What's the structure on your shirt, Mark?
What? Oh, I'm wearing the shirt from Karthik Structure.
I remember. I have it,
This
but
is,
I should have worn it too.
This is GFCC. We made shirts. We didn't make, not too many structures got into shirts. And I have a small presentation of just some pictures that I found on my computer. This is a poster of YRAM. Okay, I wanted to narrate. This is FJ, who I guess couldn't make it. FJ was an undergrad, who's now a graduate student. This is Tim, who's just graduated with his PhD from UCSF. This is VG Structure, one of VG Structures, I think. This is Karthik when he graduated. And this is another LPOA poster. These are the wine bottles, and they actually, some of them faded, but I actually have photographs of them. And there's Tom with the two kids. This is Dan Hume on the right. He was actually the very first person in the lab. There's Craig and
Kathy and
yeah, that was Craig. Let me see. Can I go back? Yeah,
Kathy and
that was
Brian
Craig, Kathy Drennan, and I don't remember this guy.
Brian Gunther.
Right, Brian Gunther. And this is at the synchrotron, and Craig's hard at work here, collecting data, I assume. And this is Heidi Schubert, who was a graduate student. And Craig, and there's G and Na. And this is Alex Smith, who worked with Tom Transon. And there's all of you at some meeting, though.
It was Montreal.
It was Montreal? Oh,
Yeah.
that makes sense, because there's a picture of a statue next, I think. So this is Bart and Craig and Tom and me and G and there's Jeannie. I thought she would be able to come. And this is David.
No, Mark Hilgers.
Yeah, Mark Hilgers. And this was who?
Lindy Dixon.
Lindy Dixon. Craig, you have a great memory as always. Let's see, I think there's one more. Is that it?
That's it.
Yeah, that's it. Okay, so let me get out of there and back to here. So
Wrap it up.
I think we're going to wrap it up because the time has proceeded. Thank you for all the wonderful comments that everyone made. I really appreciate them, and I really value your contributions to the lab when you were in it and the time that we spent together. And I'll miss everyone. I definitely miss having people in the lab, that's for sure. So any other comments? Anyone wanted to say anything? No?
Keep in touch.
So if we could keep in touch, would be great. I now have a database of all the emails if anyone's interested. So wonderful. Thanks for coming out this afternoon, and stay well, stay healthy, and hope to see you all again soon.
Congratulations again, Mark.
Thanks a lot.
Thank you, Mark.
I want to say,
Congratulations, Mark and family.
Congrats, Mark. Keep
Sorry,
in touch.
Tom, what did you
Bye.
want to say?
just really quick, thanks to Cindy for sharing Mark for so many years.
Thanks, Tom. Very, very, very true. Okay.
Oh, thanks, Mark.
Congratulations.
Thanks.
Yeah. I remember the shirt. I didn't want to soil it, so I have preserved it. I don't wear it often.
You haven't worn it at all. Okay.
I mean, I wore it. I wear it for special occasions, but I want to preserve it, so I don't want to. I just wear it for special occasions. I'm very happy to see that. I'm very happy to share this memory with everyone here. I remember Katie and Brahmara. I forgot to mention. I didn't see it on the grid view, but I remember Lisa Glass and. Yeah, there's some fun memories. I know we want to wrap it up, but yeah, it's just painting the rock in Michigan. Yeah, we had a fun time with Austin and everybody. I'm very happy. I put down memory lane like that. Yeah, thank you.
Okay, guys. Bye-bye.
Bye.
Bye.
Bye-bye.
Bye.
G is still there. Good night, G.
Good night, G. Get some
Yeah, night, G.
Later.
I hope the my gift would reach you this week or maybe
Well, that was very
next
sweet
week.
of you.
Okay, it
We'll
would
look,
be
we'll
delayed.
look
a bit.
for it. We'll look for it. Yeah, great. Thank you.
Okay.
Bye, PG.
Bye.
Vicky,
Bye,
You
Craig.
sent,
Bye,
it
Vicky.
you sent it to my home address, right?
like we saw
Yes.
years ago.
I know, lots of great memories.
Yeah. it
We,
was
we will
fun.
never
It was a
have
lot
a
of fun.
people like you in our life. It
Yeah.
was so good.
Yeah, thank you.
It's
It was
really
fun.
good. Take care.
Good luck to you, BG.
Good luck.